Thus, live streaming right now. Et puc demanar un favor? Pots mirar al teu ordinador si l'emissió de YouTube funciona? Vas al meu Twitter i hi ha un link. És el primer. El segon, després de que sí. Primer, aquest, aquí. Pots clicar? Sents alguna cosa?
Some interesting background behind this meeting. And there's also many people that we want to thank, so I'll just get right into that, and then we can keep on time. First thing I want to do, though, is apologize to our colleagues and friends in Asia and Australia, particularly because we immediately had a difficult decision to make with this meeting about the time zones. The Earth is, of course, a sphere, and there's nothing we can do about that, despite people that believe it's not a sphere. And it was a worry right from the start how to deal with time zones, and we just decided to pick time zones that worked for the 3 meeting participants. And the hope was that there would be future meetings, maybe reflecting different subdisciplines or maybe reflecting different time zones, and we'll say more about that in a second. But we did have some registrants from China, Japan, India, Australia, so I'm glad to see that some people made it, and I salute your ability to join a meeting in the middle of the night, and I urge you to think about trying to do one of these yourself. It's been an interesting experience. Having said that, I also want to thank the other organizers. They've all put a lot of work into this. There's been a lot of test meetings and technology to work out that none of us was very familiar with, and they both have young children, unlike me. I have teenagers, and so I recognize that teaching, and this is a stressful time to agree to do something like this, and yet they were both happy to take part in this as well, and it's probably more difficult for them than me, so I really appreciated that. And I'm just speaking on behalf of all 3 of us here, just so we can save some time. I want to thank the speakers. This is actually, I've organized many meetings, and I think this one has had the highest success rate of speakers accepting the invitation. And so I think that's fantastic. It's a bit of a leap of faith to try to do something like this because none of us knows how well this is going to work. And so thank you to the speakers for making the time and the effort to join us and see what happens. And we'll try to make this as smooth as we can. We also have 3 helpers with us today who are co-hosts as well. There's Mawash, Naomi, and Raquel, and they're going to be recording the meeting and helping to manage the weight room and so forth as well. So thanks to them, and they've also helped with test meetings and so forth. And lastly, the scientific societies, ISEP and ISEP, and also the PR2 database, all very generously allowed us to use their mailing lists to send this notices out to the field. Because we're not a society, this is not a society meeting. We're literally just 3 people that kind of said, hey, let's organize a meeting and see what happens. And without their contacts with the whole society, obviously, we wouldn't have got out to very many people. And I thought it was really nice of them to make an exception to do something that they thought might represent the whole field as opposed to representing the interests of the society by itself. And so that was fantastic, and we really appreciate that. Lastly, of course, the participants. This has been a really interesting experience. I had to go through the registration list and accept everything manually, and I was actually kind of dreading that, but it turned out to be a lot of fun because I saw lots of familiar names and lots of people I don't know. And I also saw a little bit about where everyone's coming from, and that was interesting. Obviously, the time zones working against Asia and Australia, but we saw lots and lots of evidence that that this format does not have a north-south bias. So we have many colleagues joining us from South America, Africa, as well as thoughts from Mexico, US, Canada, and various countries in Europe. So that was really interesting to see. And I think this is maybe one of the more international meetings I've ever been to. Then I also wanted to very briefly say a couple things about why we did this. It's obvious that we did this because there's a big gap. We're all locked up in our houses, and all scientific meetings are essentially canceled. So that's just like pretty clear cut. But also, we wanted to do this as an experiment to think about meetings of the future, because we all travel too much. There's a lot of scientific meetings. I've been thinking about this for quite a while. I actually was realizing this morning I proposed to do a meeting like this 10 years ago with the Canadian Institute for Advanced Research. And they said no, because at the time it was more expensive to do a meeting like this than it was to travel. And so times have changed. And this kind of thing can now be organized by, just three guys that kind of go, hey, let's do a meeting. So I think we should actually pay attention to that and maybe use this technology and use these ideas in the future, even when we go back to traveling. There's no reason, for example, why a scientific society can't do this every second year, and every second year do an in-person meeting. Or maybe more realistically, we should sort of put this as a challenge to societies to think, how can you add this to your regular meetings? Can we have, for instance, a cheap registration for people who can't travel or don't have the time to travel, where they can attend a meeting electronically as an option in registration? I think you'd see a huge uptick in the number of students and postdocs that were able to, go to a meeting. So we can advocate for this if this works well and if we like it. So that's one of the other reasons I was really interested in doing this. And in light of that, I actually wanted to make an announcement too, because I'm actually quite hopeful that this format will actually become popular. And one of the reasons is because I'm happy to be able to announce that this, excuse me, that this is going to be the first of this kind of meeting and not the only one, because we already have a team of people who are starting to assemble that want to carry on with this ESOP style meeting. And specifically, Philip Husnick in OIST in Japan is going to organize an ESOP-like meeting for the Asia-Pacific region. And he's putting together a group of people to organize it right now, and he's going to be asking for submissions from students and postdocs to give talks at that meeting. So I hope we see that happen very soon. And I'd like to thank Philip for offering to do that. And we'll probably be transferring all of the tools that we work out here to them so they can improve on them and carry on. So that's great news. And we'll give a little reminder about that at the end of the meeting as well. Lastly, really quickly, I just want to remind everyone of some technical things. We have 239 people in this meeting right now, which is a lot, and none of us has done this. And so we've come up with a few rules. The first rule, speakers, please keep to time. We don't have a good way of subtly telling you that you're running out of time, and so we're going to have to kind of rudely interrupt and say, you have 3 minutes left or whatever. So none of us really wants to do that. If you can make sure that your talk is timed and that you keep to time, that would be great. The audience, number one rule, keep your microphone muted, please. We have a mute all option, but I don't really want to use it. So keep yourself muted unless you're asking a question. Ideally, probably most people should keep their cameras off. I think some of us, of the co-organizers, maybe we can keep our cameras on a bit just to give the speakers a little feedback so they know there's someone there. But the vast majority of us should turn our cameras off. During talks, after talks, when it's time for questions, if you want to ask a question, use the electronic raise your hand function. Okay? If you open up your participants tab at the bottom, you will see a long list of participants. When you raise your hand, you will jump up to the top of that list or just below the co-hosts. The co-host or the host that's doing this part of the meeting will address each person in turn in the order that they put their hand up in and invite them to ask a question. They will unmute their microphone, and then after your question, you will be remuted, and you can put your hand down electronically, and then you will go back down in the list, and then the host can carry on. So if everyone can please do that, that will make everything run a lot more smoothly. And lastly, for the participants, 240 now, please just be patient with us. We're all learning how to do this, and we're going to try to make it go as smoothly as possible. Please take part in the coffee break. I think it'll be an interesting experience, and we're trying to learn some things about this. And lastly, at the end of the meeting, we might send out a little survey to see what you liked about it and what could be done better, and that will help future meetings. So if you do get that, please consider answering it. All right, so with that, we're going to move on to the first speaker, and that's Inaki Roos-Trullo. And if Fabian can unshare, perfect. Inaki, can you please share your screen? And unmute your microphone.
add something in the computer. I'm sharing now. See if you see it.
It looks good.
Perfect. Okay, so good morning and good afternoon and good evening, everybody, and thanks Patrick and Fabian and Javi for inviting me for this conference, this first online Protist meeting. I feel like at home, actually, literally, because I see a lot of faces here that I know, also the organizers, and it looks really like being at home. So anyway, I'm going to talk today. Actually, so today it's an important day for Mexico and Mexicans. So I'm going to dedicate my talk to Mexico and to the Mexican people. Not only because it's an important day for them, but also because I really believe that of all the eukaryotic organisms, if there are ones that can be defined as Mexicans, these are for sure the unicellular holozoans, so the unicellular relative of animals. And during the next 20 minutes, I'm going to try to show that to you. But let's start with the beginning. So what are we showing? So what are we celebrating today? So today we're celebrating May 5th, Cinco de Mayo, which is actually a very peculiar Mexican-American festivity. It's actually more celebrated in the States than in Mexico. And a lot of people in the States believe this is the National Day of Mexico, which is not. What we are celebrating, or actually what Mexican people are celebrating today, is the defeat of the French army by the Mexicans on the 1st of May, 1862, in what is called La Batalla de Puebla, the Battle of Puebla. And historians, they like everything that looks very epic. So the way they dress this historical event, they dress it in a very epic way. So basically, they explain that the French army was much more superior technologically than the Mexican one, but still the Mexicans were able to defeat them, probably they tell because of their bravery. And it's probably all true that, but I think there is a lot of other considerations that we may take into account when we analyze these events. Probably the knowledge of the terrain, environmental conditions, the food, and many other things. But trying to be epic is something that is very human, and it's the same problem not only with historical events, but also with other disciplines such as science. So when you try to analyze historical events in biology, you can always have this kind of explanations. Even, for example, when you try to understand the origin of animals, which is the goal, or actually, which is what we want to understand and we want to address in our lab. About the origin of animals, there have been two main problems to address it. First one is this pervasive animal-centric view that we researchers have. The second one is the lack of data for many years. So basically, we researchers, we think humans are very unique, and they are. They have a huge diversity of body plans, exceptional behaviors, and of course, an incredible embryonic development. They are unique, but they are not alone, which is something that was considered for many years also. If you look for many researchers, like animals were something like unique, fungi and plants and other protists were something separated from animals. Then later on, they realized that animals are not unique, and actually they are closely related to a group of protists known as guanoflagellates, and actually they share a not so far ancestor with fungi also. This provided a way to analyze the origin of animals in which guanoflagellate was taking a major point here. So guanoflagellates were the sister group to animals, and also guanoflagellates were considered to be homologous to one specific cell type of sponges called the choanocytes. These two views, that animals were quite unique, that guanoflagellates were the sister group, and that they shared this homology, were the basis for the guanoblastea theory, in which guanoflagellates were the ancestor of animals, guanoflagellates were eventually provided into more complex guanoflagellate colonies, and suddenly there was an important gene innovation just at the origin of animals. we was also correlating pretty well with the fossil record data that show a Cambrian explosion or an explosion of body plants at the origin of animal, suggesting an important genetic innovation and the origin of the animal genetic toolkit. However, we have learned a lot in the last years, and we know that the story is not as simple or as idealistic as that one. And the first thing that made us change this or change the perspective is that the realization that guanoflagellates are not the only lineage that is closely related to animals. It's not the only unicellular lineage that is closely related to animals. We have at least 3 additional unicellular lineages, and this is very important. They are all very different from guanoflagellates, and they are all very different one to each other. So let's revise all these unicellular lineages. The first ones are the guanoflagellates, they belong to animals. They are flagellates, protists that eat bacteria, and they can form colonies like clonal division. The most studied one is Salpingoeta rosetta, and as you can see in this standard video, they divide by clonal division, and they form rosette colonies. We also learned very recently, a few months ago, that there is a new species of guanoflagellate named as Guanoeca flexa, described by the lab of Nicolkin, that has been able to modify, that has been shown to be able to modify their shape in a very rapid, coordinated manner, actually in response to light changes. It's so rapid and coordinated that looks very similar to what happens during animal gastrulation. Besides guanoflagellates, we also have the phyllosterians, the sister group of guanoflagellates and animals. So this group here, phyllosterians, which are amoebas with phylopodia that are able to form aggregate structures or multicellular structures by cell aggregation. The most studied one, there is only 4 known species so far, is Capsaspora ozatharki. It has 3 different life stages and culture conditions. This is a timelapse video of the phylopodial amoeba one, which is the growing stage. It has a cystic stage, in which the phylopodial are closed or retracted, and then it has an aggregative stage. So the cells will eventually actively aggregate and form temporal multicellular structures. So this will be the phyllosterians. Then we have 2 additional ones, the Diasporians and Corallopithecians, that they develop through a xenocyte development. So the Diasporians, most of the Diasporians has been identified inside animal invertebrates. One of the better studied one is Spiroforma artica, which was actually isolated from inside the guts of the amphibod Gammarocetosus. The way Diasporians develop is by a xenocyte. So they start with 1 cell and 1 nuclei. The cell will start to grow, and the nuclei will start to divide without cell division. So it will have a multinucleate structure, and at some point the nuclei here will form each one its cell membrane, and then the external cell wall will break, and the cells will be released to the media. This is shown in this time-lapse video here. If you follow the black arrow, this is the one that you have to look. It's already a grown Spiroforma artica cell with a lot of nuclei. Now the nuclei, you can see here in whitish, will go to the periphery. They will cellularize, and eventually the cell wall will break, and the cells will be released to the media. There are other Diasporians that have an additional life stage or cell type, which is an amoeba stage. So once the multinucleate breaks, or the cell wall breaks, it will have an amoeba stage that will go and create the new creolimas. It has also been shown recently that during the xenocyte development of Spiroforma artica, there is a moment, one stage in which there is a polarized layer of cells that resembles quite a lot an epithelium. Then we have Corallopithecian, which only 2 known species. This is Corallopithecian limathisporum. These organisms, which is free-living marine, it's a tiny free-living marine organism, can develop either by clonal division, sometimes through a tetrad-like division, or through a xenocyte. It looks like it's able to decide whether which way it can go. So the question here is how animals appear. And in order to understand this question, there are 3 important points to understand. First will be how was the unicellular ancestor, then how was the first animals, and then once you know both of them, how the unicellular ancestor became the first animals. In our lab, we are specifically working towards this part of the question. How was the unicellular ancestor? How this ancestor became the first animals? Logically, following the theories of the Cinco de Mayo, a lot of researchers before were assuming the unicellular ancestor was pretty simple, both morphologically and the cell behavior and genetically. So the first question we had is whether that was true, whether how complex was the ancestor, was really as simple as previously believed or not. And the only way to do that is by comparative genomics. So in the last 10 years, between the lab of Nicole King in UC Berkeley and our lab, we have been able to obtain the genome sequence of several of these unicellular relatives of animals, several choanoflagellates, 2 phylasterians, several ectosporians, and 1 Protistologian so far. And the data is showing us that the unicellular ancestor of animals was relatively quite complex, much more complex than previously thought. To show that, I'm showing here a picture of the genes and gene families and pathways that were thought before our data to be specific to animals, because they were not present in fungi or in any other organisms. Several signaling pathways, several addition pathways, and several genes involved in transcription and regulation. After the genome so far of all these unicellular holozoans, this is the view that we have now. There is so there is few genes and pathways and gene families that seems to be specific to animals. And this can even change if we sequence additional taxa. This is an important point. More taxa we sequence, the better perspective or the better reconstruction we can have in the cellular ancestor. And every time seems to be more complex. Something similar to what happens to later. I'm going to use 2 examples just to show how important it is to have different taxa from the unicellular holozoans in order to reconstruct the pathway, the evolutionary pathway of genes towards the origin of animals. This was the view of integrin addition, which is a very important integrin addition system of animals before, if you only compare with choanoflagellates. So integrins were thought to be specific to animal, originated just at the origin of animals. When we have the genome sequence of other unicellular holozoans and other eukaryotes, the perspective changed dramatically. Integrins were present before the diversion from opistocones, and they were secondarily lost here, the crosses. So if you compare only with choanoflagellates, NF-kappa, ETS, SMAD, nuclear receptors, Greniket, T-Box, or Brunis, were thought to be specific to animals. Once you have more taxa, then the story changes dramatically. And if you sequence more choanoflagellates, this also changes dramatically. Being a lot of those genes, transcription factors, originated before, and some of them, the crosses here in color, secondarily lost in choanoflagellates. So we know that the unicellular ancestor of animals, we learn it's genetically quite complex. So the question is, what are those metazoan genes, so genes involved in the multicellular function, doing on those unicellular taxa, and how were they co-opted? I'm going to use just one example because we have not been able to develop this much further, even if we are extremely interested. The example I'm going to use is brachyury, which it's a gene, it's a transcription factor that in animals is involved in gastrulation. And we discovered there is a brachyury in the genome of Capsaspora of this phyloprotected amoeba. If we try to see whether this is conserved with the brachyury of other animals, we observe that it's very conserved. So this is a normal Xenopus, normal embryo, and brachyury is working, gastrulation is working. All these lines here, this color is merely one of the downstream genes of brachyury, so everything is working. So this is a double mutant of brachyury, of Xenopus brachyury. There is no brachyury, there is no gastrulation, there is no normal embryo. If under this double mutant you inject the Xenopus, the endogenous Xenopus brachyury, then you recover the function. You recover the normal embryo, there is gastrulation, and brachyury is working. Downstream genes such as those ones, here, are working. And the same will happen if instead of injecting the endogenous Xenopus brachyury, we inject the Capsaspora brachyury. Again, we have a normal embryo, activation of downstream genes of brachyury, gastrulation was working. However, this is not telling so much about the function. So in this case, we did a ChIP-seq of Capsaspora brachyury, and we analyzed the downstream genes. And we observed that among the downstream genes, they are enriched in functions involved basically in cell motility, which is the same if you analyze the genes, downstream genes of mouse brachyury. So there is an homologous, some kind of homology between the transcription factors. We can even say that there is a regulatory transcription factor network which is conserved between Capsaspora and animals. However, to know more about the function of the genes, we need genetic tools, and I will come back to this later. So we know the unicellular ancestor of funny was genetically quite complex, and the co-option played an important role in animal origins. So the question is, do these unicellular taxa also have genomic regulatory mechanisms such as those in animals? In order to address that, we work with Capsaspora photography because of these different life stages and because of its genomic annotation and its phylogenetic position. And what we observe at this stage, these life stages are completely differentiated at the transcriptional level and the proteome levels. For example, this will be the proteome. Each life stage have different proteomic profiles, including associated to specific protein functions, the same as happens if you analyze different cell types in animals. Not only that, if you analyze the phosphoproteome, then you see that there is also phosphatide, differential phosphopeptide abundance depending on the cell of the cell type of the cell or the life stages, similarly to what happened also in animals. Not only that, but if we analyze specific transition factors that in animals are key for animal development, like p53 or runis and kappa, they have phosphopeptide sites, signaling sites that are differentially phosphorylated depending on the life stage. So here green will be more phosphorylated in the amoeba stage, blue, red and blue will be more phosphorylation in the cystic, and orange will be less phosphorylation in the aggregative states. So basically, Capsaspora uses mechanisms such as histone modification, long non-coding RNAs, differential alternative replacing, or differential phosphorylation of transcription factors, which are the same mechanisms that animals use. So basically, Capsaspora uses those mechanisms to transition from one life stage to another, similarly to what happens when animals transition from one cell type to another cell type, or differentiate into one cell type or another. So the unicellular ancestor of animals was genetically quite complex, and it had animal-like mechanisms of cell differentiation. Another question that we may wonder is whether there are more unicellular holozoans yet to be discovered. And yes, there are. So very recently, for example, the lab of Patrick Killian just showed that there are additional phyllosterians that had just discovered Pigoraptors and additional corallitrians, Corallochitrian, which are also morphologically very different from the ones we had before. Not only that, but if you analyze metabarcoding data, as we did recently with the Tara Oceans data, we observe that there is a lot of novelty inside holozoans, both specifically within conoflagellates and within ichthyosporans. Not only that, but we were able also to analyze, I cannot explain now with more detail, but you will have to trust me, everything here with in red are different otus that represents a completely different lineage from the other holozoans that we know today, and we call them as marine small holozoans or MASHO. So I believe that these new organisms will become very relevant at some point to understand the animal origins and the evolution of holozoans and eukaryotes. One of the most fascinating things about Mexican culture for me is their richness and complexity, how they can mix reality with fantasy, the colors, also the vision for both life and death, monster, angels, or Catholicism with paganism. And I think this resembles quite a lot what we see in holozoans. We have holozoans, or the holozoan clade, is an amazing clade. It's an amazing clade with an assemblage of complex and simple cell behaviors. So we have cell aggregation, xenocytes, complex embryonic development, and different lifestyles from parasitic to eukaryotic predators, so bacteria predators as well. So If you want to understand, for example, and I'm finishing, Patrick, don't worry, in 1 minute more or less. If you want to understand, for example, the origin of animals or how protists became animals upon, you have to go to holozones. If you want to understand how such a complex embryonic development of animals could eventually came from an ancestral morphogenetic movements present in ancestral organisms, then you should go to animal, and you should go to holozones as well. If you want to understand the evolution of cell cycles, cell aggregations, or cell division, or the regulation of complex life cycles, I think one of the best places to go in the tree of life is again holozones. For example, if you want to analyze cell division, within holozones we have a normal clonal division such as Capsaspora. We have the sinusoid division, multinuclear division of Diosporians, each one which is a specific peculiarity. And we have organisms as Corallotrichium that seems to be able to perform different kinds of cell division. But the most important thing, and I'm finishing here, is not only that we have this splendid group of organisms, but now we have the capacity to analyze those questions in another level, experimentally. So we have done a lot of effort in the last year to try to develop genetic tools in these organisms. So for example, the lab of Nicole has already developed transfection and CRISPR in guanoflagellates. And in our lab, we have been able to develop transfection in one phyllosterium, Capsaspora, in 2 different Diosporians, and in one Corallotrichium, in Massispora. I think this provides the scientific community with a unique functional platform to understand not only animal origins, but also the evolution of cell biological features. So I encourage any of you interested in any of those questions, feel welcome to use these organisms, and we will be very happy to also to provide feedback and help if you want to do that. We are not Mexicans. The lab is in Barcelona, but I can tell you, we love the Mexicans of the Tree of Life, because we have seen in the last years that any bit that we learn about these tiny organisms, it's a big, better understanding, not only about eukaryotic evolution, but also about eukaryotic biology. And with this, I finish here. I want to acknowledge all of my lab, all the funding agencies, and of course, all of you for listening. Thanks a lot, and I'm glad to take any questions.
Right, thank you. So you can raise your hand if you want to ask a question. Let's give people a minute here. Okay. Elizabeth, I will unmute you.
Hi, hello.
Yep.
Hi, okay. Yeah, so thanks, Iñaki, for doing the talk. It was great to see you show. I was wondering if you say you're seeing this big diversity of Diosporians in this meta-barcoding network analysis. You think that those are free-living species or free-living stages or dispersal stages? So what is your idea about it?
Yeah, it could be either way. So I think we cannot really answer it. By my guess would be they are either free-living or they have a free-living stage. Actually, for most of the organisms that have been originally described as parasites, we have been able to see in the last years that they are not parasites, or if they are, it's only a temporal life stage. So my guess is that a lot of the things that we are seeing are free-living, or they have at least a free-living stage.
Okay, so this opinion is also slowly changing, yeah, because I haven't been following this for a while, but it seemed like this is not, not everything's a parasite, what we saw is, and this seems to confirm that now if you see this diversity.
Yeah, the thing is, until now, most of the holozones that were identified were isolated from animal invertebrates because the researchers involved or interested in animals, they were open the animals and finding the organisms. But I'm sure, and actually you know that if you go to the environments and you check that in the environments, then you can also find other organisms.
Yeah. All right. Thank you.
Thank you. I'll ask a question. just while we're waiting. But first, I want to encourage people to put your hands up and ask questions. I know it's a little intimidating to figure out how to do this, but down at the bottom of the participants bar, you should see a raise hand icon. So please go ahead and do that. I was actually interested, and I have to admit, I'm trying to organize a lot here, and I'm a little distracted, so I apologize if you've sort of already covered this. But do we have a good sense of how many new lineages of holozoans that we haven't actually characterized there are out there from the molecular survey data? I know that it's been covered by Javi not that long ago, but it looks like you're saying that you're finding new lineages still that we don't know what they're like. So how much of the diversity is still out there to find, do you think?
Yeah, well, I don't know. I mean, the short answer is I'm not sure about that. But my guess now from the analysis we did in the past with Javi and the new analysis we are doing, is at least there will be 2 or 3 additional lineages. This is also, we are entering into a nomenclature thing, because if we find a lineage that is sister group to some of the lineages that we have, should we consider a new lineage or not? And I guess it also depends. Once we identify them and we describe them, that will help us to define whether it's a new lineage or not. But my guess would be that at least there will be 2 or 3 additional lineages, and by this I mean different from the ones I described at the beginning of the conference.
I guess I also wondered if we're systematically looking for certain kinds of organisms, and we're actually overlooking the tiny little not very distinctive monoflagellates that might make up a lot of the diversity. Sort of what I was getting at.
Exactly.
Okay, are there any more questions from the audience? If you have a question and you're struggling to figure out how to raise your hand electronically, you can also hit the space bar to unmute yourself and announce it if there's no other questions in front of you. But I see Fabian put his hand up.
Yeah, sorry, for some reason I can't raise my hand electronically.
Co-hosts can't, so if you have if you're a co-host and you want to ask a question, you'll have to do it manually.
So thanks, Iñaki. It was really interesting. So you said that a lot of these genes, animal-specific genes, now are found deeper and deeper in the tree. But at the same time, if I saw it well, a lot of these genes are actually lost on the choanoflagellate branch. Is that right? If it's right, does this relate to some specificity of the choanoflagellate lineage? How do you interpret that these genes, originated quite early on in evolution, but then they were lost on the choanoflagellate lineage?
Yeah, there are 2 issues here. So the first one is that there were only 2 choanoflagellate species genomes analyzed when those analyses were done. Things work this way. Apparently, those 2 choanoflagellates are the one that probably lost more genes compared to other choanoflagellates. Now that later on, more choanoflagellates transcriptomes were sequenced. So that's something to take into account. So for whatever reason, those 2 specific choanoflagellates have lost more genes. But also, maybe some specific taxa, and that could be these 2 choanoflagellates or maybe other choanoflagellates, maybe they have reduced the capacity of performing other cell behaviors. And by reducing them, they have reduced some of the genes or the genetic toolkits that they needed to perform those. So I think a lot of that has to be with that. So I will imagine that if there is an organism that needs to be into completely different environments, it will need more genetic toolkits to perform different cell behaviors, depending on the cell environment or of the environment of each moment. But if there is an organism that has a very specific environment or also a very specific life stage, like the lifestyle of choanoflagellates is very specific, then maybe some of the genetic tools that you could have or the ancestor had are lost because they are not needed, and you are better fit for your specific ecological needs.
Great, thanks.
All right, thank you, Inaki. I think we're good. Can you please unshare your screen? Thank you. And our next speaker is Ross Waller from Cambridge. Ross is there. Excellent. Ross, can you please share your screen? And Ross is going to talk about the subcellular localization of proteins in toxoplasma. All right, that looks good.
Okay, good to go. Good.
Thank you, Ross.
Thanks very much. It's a pleasure and an honour to be experimented on in this way. So I hope it goes well. I think this is a great idea. So I'm very grateful for the organisers for getting this together. So I want to start off with some pictures of fish, oddly enough, but I really love getting under the water and looking at fish. And one of the things, the real pleasures of this is the diversity that you see in this group of organisms that are otherwise all related. And it can really be sort of baffling to try to reconcile this. And I think there's some nice examples here. In particular, the frogfish down at the bottom right-hand corner here. If you looked at this, I think you'd be forgiven for not realising it was a fish in the first place. And it's got these legs and it moves around at the bottom of the ocean. And indeed, the question sort of strikes me, are these new features or are they derived from some feature that's common with fish? And indeed, these are the fins of the fish. I mean, similarly, there's this angler here, the lure, that's on the nose. And I was always puzzled by this, but again, it turns out this is a modified spine of the dorsal fin. So with a little bit of extra knowledge, you can actually reconcile these things and understand how this sort of diversity has evolved. But nevertheless, I think organisms' morphological and structural diversity can be really hard to interpret. And of course, this extends very much to protists as well. Here's a collection of protists here, a lot of them are dinoflagellates, and again, sort of baffling diversity here. And again, it's a real puzzle sometimes to figure out, A, if they're related to each other, and B, how has this diversity arisen amongst these organisms? And perhaps a few of you will have keen eyes and you'll see that I've snuck an apicomplexan in there as well. And indeed, here's an apicomplexan on the right and a dinoflagellate on the left. So these organisms are related to each other, but if you were to look at just the surface detail here or the morphology, you wouldn't ever really know that. There's nothing much to connect the two. Even if you look inside these organisms, there's a lot of complexity in here. And I guess we look at these images and we're drawn to the things that we understand, but there's a lot of detail here that we really don't understand at all. And we can try to simplify this and sort of point out the things that we do understand. And indeed, there's a lot in common between these organisms. They've got common organelles we recognise. And indeed, in dinoflagellates and apicomplexans, they have this system of alveolar membranes, what's called the inner membrane complex, the IMC in apicomplexans. And this is a unifying feature. And indeed, it extends to the ciliates as well. So this groups the alveolata together. But there's a lot of other detail here that's kind of still very hard to interpret. For instance, around the flagellar pocket region here, or the flagellar base, there are all these vesicular structures and other structural elements here. In apicomplexans here, again, there's structure and there's vesicular compartments all around the apical complex, which is the invasion machine with the apicomplexans. And it leaves these unanswered questions in my mind. Do these structures have anything in common, or are they novel and not shared between these organisms? So really, I think as evolutionary protistologists, which I know a lot of us in the audience are, we're often trying to understand or ask the question, how can these differences be explained? And is it because there are processes of novel structural formation? Are some of these structures new? Are they divergent from a common origin? Have they been repurposed in some way? Have some been lost? There are a number of evolutionary processes that might lead to this diversity. But it's actually a really hard question to try to get answers to. So, of course, genomes have been fabulous tools for capturing the molecular complexity of cells. And indeed, these have propelled a lot of the work we've done in the last couple of decades. But if we look at genes in common in this case here with Toxoplasma and Plasmodium, two apicomplexans with their human hosts, there's actually not very many genes that are common to them all in the context of their full genome. Toxoplasma here are a minority compared to genes that are unique to each lineage. So this reflects the fact that these cells are complex. There's a lot of diversity, a lot of novelty. And indeed, if we look at Toxoplasma and Plasmodium, even the openly acknowledged genes, about half of them are just hypotheticals. We know nothing about them in Toxoplasma, about a third in Plasmodium, but in reality, we actually don't know much about a lot of the complexity of these cells. So what we'd really like to do is ask the question, what's the cellular context of all of this proteomic and genomic complexity, and try to reconcile the novelty that's in these cells with the structures and put some context to their properties. So when I joined Cambridge, I had the pleasure of meeting Catherine Lilly, who's a colleague in the department. And pleasure because she's a lovely person, but she'd also developed this tool called Hyperlogic, and it simultaneously captures the steady state cellular location of thousands of proteins at once. And when she told me about this and showed me her data from mammalian cells, I really, my mind was blown, and I was immediately fantasizing about what we could do if we could apply this sort of technology to some of our protists. So we got together some money along with Anna Payne, and we managed to hire Constantine here. So this is a picture that Constantine has a pretty sharp suit. He should smarten the place up a bit. But in fact, that's the last time I've seen Constantine in a suit. So despite being a little bit dishonest about his dress code, it turns out that Constantine's actually brilliant. So he's driven this project I'm going to describe to you now. And indeed, we've just released this in a bioarchives report, so I'm going to skim over the detail of what he's done, but it's been a major effort on his part. And he's taken toxoplasma cells, an apicomplexan, he's pressurized them to about 2,000 psi with nitrogen, released that pressure. I think if you're a diver out there, you'll appreciate the headache that you get from that. And indeed, this is toxoplasma with the headache of all time. It's been broken open and dispersed on a density gradient here. So what we're aiming to do is to somehow separate or disperse compartments of the cells according to some unique property, in this case, density. So the next step is to accurately quantify, using mass spectrometry, the abundance distribution profile of all proteins across this gradient here. So indeed, in this experiment, Constantine's measured across all 10 fractions here about almost 4,000 proteins, their abundance. And that's represented by all these grey lines here, which are superimposed on each other, so you can't really see much. But I've highlighted an unknown protein in black there, a protein that we otherwise knew nothing about before this study. So in this case here, we can see that it's sort of peaking towards the bottom part of the gradient. So we can ask the question, well, what are other proteins we know about? Proteins we know of that should be the mitochondrion, perhaps, or the inner membrane complex, that unique structure to alveolata. And indeed, we can see that in green, the mitochondrial proteins have all got the known ones we already knew about. Have got a similar profile, which is distinct from our unknown one, which is actually a lot more similar to the inner membrane complex proteins here. So what Constantine has done to give power to this sort of data set is he's done it 3 times. Got a delay on my presentation here. Okay, so he's done it 3 times, and in each case, he's actually ruptured the cell in a slightly different way and used a slightly different gradient to disperse it. So we're aiming again to capture even more complexity in these cells. And again, you can see that the unknown protein is following pretty faithfully that trace of the inner membrane complex proteins through this trace here. So we've now got 30 dimensions of data for a little under 4,000 proteins, and we can model that in a way that aims to represent proteins that have similar distribution profiles across these 3 gradients. So in this case, we're projecting this as a T-SNE projection, and proteins are clustered now according to the similarity of their profiles across these gradients. And indeed, the inner membrane complex proteins are all forming this tight little cluster down here. And this structure in the data that we can see in the T-SNE projection here is good evidence that, or good indication that we were actually seeing some proteomic structural data from these cells. So it's representing something that is actually based on the biological organization of the cell. So again, we can take a lot of known marker proteins and project those across those projections. And if we overlay those on the T-SNE projections, we see that indeed these clusters are representing compartments with proteins that we already knew about. And again, we can do Gaussian mixture modeling on these to try to assign the parameters of each of these clusters and use that to capture in a probabilistic manner any unknown proteins and assign those to those clusters as well. So the outcome of this work, as soon as it will advance, So I'm having trouble here. The outcome of this work is about 2,500 proteins that have been assigned to 26 distinct subcellular niches in toxoplasma with a cutoff here of 99% probability. So I just want to pause for a moment and acknowledge that this was a fantastic outcome. I think we were, to say the least, pretty excited by this, and it was at least an order of magnitude better than my wildest dreams when I first chatted with Catherine in the first instance. And again, as I say, there's a lot of analysis we've done of this, and you can read about that on the report that's in bioRxiv at the moment. I want to point out some of the questions that we've then been applying these data to, including some that are in the report at the moment and some ongoing work. And this is then a question that we really wanted to ask is, what can we learn, having now representative proteomes in the different compartments of toxoplasma? Can we use this to think about the evolutionary trajectory of apicomplexans through parasitism to toxoplasma? So in fact, effectively, we're looking back in time from toxoplasma, or I'm aiming to look back in time. So each of these versions of toxoplasma in the past is effectively a common ancestor across this phylogenetic range here. So back to the coccidia, or back to the apicomplexa, the alveolata, and so on. And we're asking the question, what were the new proteins that arose and are common to that common ancestor at each of those points? And where do they occur in the cell? Are there parts of the cell that have to show particular fast innovation for novelty at certain points in the evolutionary trajectory of apicomplexans? So this analysis here asks that question and presents the outcome to that. So across each of the compartments that we've got proteomic data for now in the vertical axis here, and across the x-axis here is phylogenetic depth here. So we've got toxoplasma at the left-hand end, going back to all eukaryotes on the right-hand end there. So effectively, this is telling us here that, in this case here, at the base of the alveolata, so common with ciliates, dinoflagellates, and apicomplexans, where there was particular enrichment for novelty is in this compartment here, which is the inner peripheral proteins of the plasma membrane. And indeed, this is very interesting because this correlates with a structural feature, the alveolar membranes here. And indeed, we know that in apicomplexans, this is the major site for regulation of signaling pathways for invasion. And certainly the proteins that are enriched at that point here are enriched in, at least the ones that we know we know some information about, are enriched for signaling proteins involved in calcium and cyclic nucleotide signaling. So it's giving us an indication that actually this innovation at the base of the alveolata really was, at least in part, about developing a platform for signaling in the cell. Again, we can look a little more recently here at the base of the mesozoa, so dinoflagellates and apicomplexans, and the hotspot for innovation in this case was in the mitochondria. And again, we actually do know that there's a lot that's very novel about the mitochondria that's shared with apicomplexans and dinoflagellates. Again, if we look at apicomplexans and their common ancestors with chromerids here, so this is before we think about parasitism really being the niche of the apicomplexans yet. The chromerids, of course, are photosynthetic, but believed to live in association with animals. And indeed, it's the inner integral membrane proteins of the plasma membrane. And these are highly enriched, these proteins in this case, with transporters, suggesting again that there's innovation and modification of the plasma membrane that's happening at this point, in fact, even before parasitism has really become the main activity of apicomplexans. Again, into the apicomplexans proper, and we're seeing compartments like the inner membrane complex again, and we know there's a lot of motility machinery and a lot of machinery that's involved in invasion there. And then finally, looking much more recently in the Sarcosistidae, so they're the close relatives of toxoplasma, and now it's all of the organelles associated with the host interaction. And again, we're seeing that signal very clearly here. But the nice thing about this is not that just that we get this chronology of the evolutionary events going through the progress to toxoplasma, but we've now got proteins here that we've identified, or this analysis identifies as being perhaps key to some of those innovations. And a lot of those proteins, their functions are unstudied, so we've got an opportunity to try to start to functionally tease apart these evolutionary events. So another question that we're asking is, are things that we can't see really not there? So we're getting a little bit metaphysical with this question here, but in this case, we're directing at this question, is a conoid present or not in the aconoid acida? So I knew I was going to trip myself up putting that in. But this is the class of apicomplexans that includes Plasmodium. So is Plasmodium and other members of that class, do they truly lack a conoid, which has been speculated for a long time? So the conoid is best known, probably best visualized in Toxoplasma. That's where there are the nicest and the most images. But it's this tubulin-based barrel structure that's right at the apex of the cell. It's involved in invasion. It actually is motile in Toxoplasma. It can move in and out at the end here. And secretion of invasion factors occurs from the micronemes and the rupturies through this structure here. And we know the conoid is present in a lot of apicomplexans. It's even present, or something that's very similar to it, in close relatives of the apicomplexans, so the chromerids, coprolalids, and dinoflagellates here. And again, some examples of those shown here, involved in predation in some of these organisms. Brachyury here, again, it's got a lovely conoid-like structure here made of tubulin. We don't really know what it does with Brachyury. But within the apicomplexans is this group that's been believed for a long time to lack the conoid. And this is all based on ultrastructural and microscopic evidence here. This is an image of a Plasmodium cell here, and certainly people have noticed it. There are ring-like structures there, and some people have even labelled these as a conoid. But by and large, the consensus has been that in the field that the conoid probably is something that's been lost from this group. But as I say, this is based on microscopic data, and I guess we've really got to ask the question if that's enough to answer this question. And what we'd really like to know is what's the molecular architecture of this structure in apicomplexans, and do we find something similar in the apex of Plasmodium and others? So this is something that Ludek Kareni has focused his attention on. And indeed, in the Loppet data here, we've got a cluster of about 65 proteins that are specific to the apex of Toxoplasma. And he's set about validating about half of those so far, so he's been making reporter tagged lines in Toxoplasma and validating those by microscopy. And indeed, about 21 of those are associated specifically with the conoid in the apical complex. So here are some examples, and in fact, he finds that they're either at the anterior end of the conoid, shown in red, so the new protein is in red, a marker protein for the conoid body is in green here, or they localize to the whole conoid body, or others are localized specifically to the conoid base here. But as I say, 21 proteins have these localization patterns in Toxoplasma. But really interestingly is that 13 of those 21 have got orthologs in Plasmodium. And this really begged the question, well, where are these proteins in Plasmodium? So we teamed up with a great collaborator, Rita Tawari, in Nottingham to do this work, and her group have tagged 7 of these proteins. We know what their location is in Toxoplasma. And indeed, in Plasmodium berghei, which is a great model for Plasmodium studies because it's easy to get access to the mosquito stages, the ookinete and the sporozoite, and the blood stages as well. And we're finding that these proteins indeed are exactly where you might expect them to be if they're fulfilling some similar role to the conoid. So here's some examples here in the ookinete, the sporozoite. In this case here, this protein is present in all of these stages. Some of the other proteins are present in the mosquito stages only and absent from the merozoites, which I think is also interesting because it tells us that the apical complex is divergent between the different stages of the parasite. So what do we know about these proteins? Well, a lot of them have got coiled structures, so they're obviously involved in the structure of the conoid. A lot of them are involved in signaling. Again, we've got calcium and cyclic nucleotide binding domains. A lot of them have got kinase and phosphatase activity. And again, these are all features that we know about, or we know that are very important for regulation of key events in apicomplexans. So I think this is telling us that a lot of what we find in the conoid in Toxoplasma is probably there in Plasmodium as well, even if the structure presents slightly differently. And I think we're sort of at a new phase of being able to ask questions about the conservation of this feature across apicomplexans in more detail. Now, I realise I'm at 20 minutes. I've got about 4 minutes left. Do you want me to just skip to the end, Patrick? Actually, not hearing you, but I can do that. So look, I'm going to wrap up. But I think we're at a really exciting time here for looking at for doing global compositional comparisons across these divergent cells. And we've got data now for toxoplasma, and we're able to ask, I think, all sorts of questions about the evolution of these cells. But excitingly, now we're also in the process of expanding this. So Scott Chisholm has now got some data for Plasmodium, so we're able to start to do Plasmodium toxoplasma comparisons. We've teamed up with some other colleagues. We're doing some Armeria as soon as the lockdown is over. We've been working with Katie and Boris in Pennsylvania with Cryptosporidium. So again, I think we're getting some nice representation of apicomplexan data. And we're also working with dinoflagellates. Thomas has got some new data for Symbididium, Ilan for Brachyury, and we've got a new exciting project to start to tackle a few other interesting dinoflagellates here. So again, I think this is sort of a new opportunity to really ask new questions with new data sets across these diverse organisms. So again, just to thank everybody that's been involved, and it's been a huge team effort. Constantine has certainly led the charge with a lot of data, but a lot of the validation work and analysis has been done with other people in the lab. It's been a fantastic collaboration with Catherine and her group, Arna Payne in Cast and Rita in Nottingham, and other colleagues who are working on some work which I've just skipped over, but again, as I say, it's been a great team effort. So thanks very much for listening, and I'm happy to take questions.
Thank you, Ross. That's fascinating. Let's see if we have some questions. Yes.
Thanks, Ross, for the talk. I would just have a technical question. Were the toxoplasma cultures synchronized or not?
Yes, that's a really good question, because it is key. So these analyses used almost 10 to the 10 cells. So there's a lot of cells in there, and it becomes a global average. So the better synchronized, the better. And in this case, this was working with the extracellular stage of the tachyzoid. So they're probably relatively arrested in that stage. Constantine's starting to do work now in the intracellular phase, and that becomes much more of an issue. And we'll aim to keep them as synchronous as we can, because we'll start to lose some definition. But yeah.
Thanks.
Thank you. Zoltan?
Hello, great talk, Ross. I wondered, you showed that a lot of innovation was brought into apicomplexans with conglomerates in terms of transporters. Did you look at if these transporters were mostly on the cell surface or intracellular? I mean, if they are draining more kinds of nutrients from the environment, or maybe this implies more of a rewiring of the intercellular biochemistry.
So that compartment that was enriched at the base of apicomplexans and chromerids is all proteins in the plasma membrane. So they're all integral plasma membrane proteins there. So we would infer from that they're presumably mediating interactions with the external environment.
Interesting. Thank you.
We've got Usman.
Hello, Mr. Ross. I want to ask a question as you mentioned at the start of your talk, that you use some special instrument, hyperlobit, to understand the state of proteins. So I want to ask that, can we use this structure, this system, to understand the location and state of protein in other organisms?
Yes, no, absolutely. One of the things that's been very gratifying, and is we found that protists in this case lend themselves even better to this methodology than some of the cells that Catherine's group originally worked on. And I think it reflects the fact that we've got very sort of highly organized and fairly homogeneous or cell populations rather than a large cell with lots of versions of different structures. So it certainly can be used in other organisms. As was sort of pointed out before, one of the challenges is to have as same or synchronous a population of cells as possible, or organisms as possible. We are actually expanding this to do some work in the corals and partly trying to capture the symbiosis of dinoflagellates in the corals. But one of the challenges you start to have when you go to multicellular organisms is you'll have a mixture of different cell types. Again, you'll start to perhaps lose some resolution where there are different populations there. But I would say that it's looking like it's a really powerful tool. And I think very applicable to a lot of cell systems.
Thank you so much.
I'll ask a quick question. I'm wondering, how would you see, what would it look like if you had a protein that was targeted to two compartments? Like, would you get a bimodal distribution in two different places? And would that actually come out in the kind of analysis that you did? And did you see it?
Yeah. So that's certainly something which you can do. And if you've got a protein that's in two locations, its distribution profile will be some mixture, some summation of those two, and therefore by the sort of analysis as it's run in the first instance, it won't look like either location A or B. It'll look like a novel location, but it's really just representing the novelty of that. But one of the things that Konstantin and Oliver Crook, who's one of the bioinformaticians that's been working with us on this, have been able to do is to assign the probability of any given protein belonging to all of the compartments. And we find then that you can start to dissect, and it doesn't work always perfectly, but you can start to find proteins that have mixed locations. You'll get then distributions of probabilities across those different compartments. So we are able to dissect some of that. And again, some of that's we've included in the report.
Fabian has a question.
Yeah, thank you, Ross. It was really fascinating. I failed to see in your graph, where you showed the sort of like the tracking down the location of the proteins along the phylogeny. If you had a plastid category in that plot, and if you do you see any evidence for maybe innovation related to maybe a plastid acquisition on the branch linked to mysosome? Yeah, this one.
Yeah, so nothing stands out particularly. I mean, there are some, these are sort of lower significant hits here. And I guess I should point out here that we're only looking, in this case, we're looking where there's a skew in overrepresentation that's statistically significant. So there are certainly, proteins that are novel at this point, various points along these trajectories. But it's not that it looks like there's a huge change that is detectable now with the data from Toxoplasma. But I think we should recognise also that we're looking through this sort of period of evolution through the lens of Toxoplasma only, and it's not to say, if things have then subsequently been lost in Toxoplasma, we're not going to see those with these data, and we might when we start to include other organisms in the data from other organisms.
All right. Well, if there's no more questions, thanks, Ross. I'm actually very excited about this technology too. I just can't wait to see where this goes. All right, the last speaker that we have before coffee break is Raul. Raul, can you turn on your camera, mic, and share the screen, please? We're going to hear about Symbiodinium and coral.
Exactly, yes.
Right,
thanks for having me here. I'm very, very happy to be invited to this amazing symposium and the great talks we're having. Today I'm going to tell you a little bit about whether symbiosis can drive genome evolution of Symbiodiniaceae. And Symbiodiniaceae is a group of dinoflagellates, typically known because of the symbiosis they establish with corals and many other marine organisms. So if you take a sample of Symbiodiniaceae and put it under the microscope, you'll probably see something like this. But they do display some morphological diversity. However, their diversity is way more evident at the ecological and genetic levels. So in the ecology, we have a wide variety of forms that are symbiotic. We have some that are host specialists, some that are host generalists. Some of them occur in lower abundances within the host, which means they're opportunistic. And we also have a big range of free living species, free living taxa. Now we also, thanks to the use of molecular markers, we also learned that they also have a very huge genetic diversity, in such a grade that only 2 years ago they were all part of this genus Symbiodinium. Now they have been reclassified as family Symbiodiniaceae that comprises several genera. Now, we do know that symbiosis can affect how genomes change over time. A lot of what we know currently is based on the study of bacterial intracellular parasites and symbionts. And something, so in the early transition of intracellularity, we have this very dynamic, unstable phase where there is a lot of things moving around. There is a lot of structural rearrangements. There is activity of mobile elements, and there is gene duplication, pseudogenization. And over the time, many of these genomic elements, many genes, starts getting lost. So the genomes get smaller and smaller until they reach this very reduced, kind of stable stage of the genome, typical of obligate symbionts and parasites. Now, what about Symbiodiniaceae? In Symbiodiniaceae, we do have, as I mentioned, a wide variety of symbiotic associations. We have free living taxa, we have facultative species, and we also have some that may behave as obligate symbionts. Based on what we know from other organisms, I drew these expectations of what we expect to find in these three very broad categories. Most of the symbionts in Symbiodiniaceae would fall into this category of facultative symbionts. So we expect them to resemble this very dynamic phase of the genome. So we generated two genomes. We generated based on short-read data from Illumina and based on long-read data from PacBio. One of them is a symbiont from clams, and another one is a free-living species, and this is actually the type species of the genus. And we compare them, and something very interesting that we realized is that they were actually very, very different. This is just results of whole genome alignment. And we can see that very little, about 1.3% of the sequence from the symbiont aligned to 1.5% of the sequence of the free-living species with high similarity. And this was, as I mentioned, based on whole genome alignment, but this was also confirmed just mapping the filter reads to each of the genomes. So we wanted to know what was contributing to this divergence. And something that we also noticed was that the genome sizes were quite different. So the size of the symbiotic species was way larger, almost twice as large as the genome of the free-living species. So we decided to use this as reference to see different types of genomic elements that were contributing to this difference in genome size. So this is just a ratio of the sequence covered by different genomic elements between these two species. And something that really sticks out, some of them that really stick out are these different types of duplicated genes and repetitive elements that, again, is kind of what we expect from the genomes of facultative symbionts. And, oh yeah, I forgot to mention that these repetitive elements are actually associated with mobile elements with transposons, again, what we expect. So this is just showing a bit more in detail these repetitive elements. This type of plot is known as a repeat landscape. So on the x-axis, we have some measurement of divergence from an original sequence. And on the y-axis, we have the fraction of the genome that they cover. And something that I would like to highlight here is this peak at very low divergence, very conserved, that suggests, again, because these are associated with mobile elements, that these are functional transposons or retrotransposons. And we can see that this peak is actually way larger in the genome of the symbiont compared to the genome of the free-living, again, matching our expectations. I also mentioned before that different types of gene duplications were contributing to these differences between the two genomes. So we expected to see gene family expansions because of this more increase in the genome of the symbiont compared to the genome of the free-living. However, it wasn't really the case. So what I'm showing here is just this plot where we show on the x-axis the gene family size ratio between the two species. If it's expanded, in the symbiont is in pink here on the right side of the graph. If it's expanded in the free living, it's here on the left side of the graph. The y-axis is just significance. And what we can notice here is that the two sides of the graph are quite symmetrical, which means there is not really big difference in gene expansion in the symbiont. And we were wondering why is that? And the answer probably lies on the fate of duplicated genes. So if the gene copies, if the function of the gene copies remain about the same, then the sequences are gonna be basically the same. They're gonna have little changes, and then that would contribute to gene family expansion. However, if the function changes drastically, the sequence starts becoming dissimilar, and they would probably be classified as a different gene family. And some evidence of this that we found in our genomes was in the number of single copy genes. So they were more abundant in the genome of the symbiont species compared to the free living species. However, when we look at the protein domains encoded in these genes, we notice that they had very similar profiles, which probably means that these are very highly diverged homologs that are just too dissimilar to be closer in any gene families. Another possible fate or destiny of these gene copies is just loss function. And in this case, the sequences generate quite quickly. Obviously, they wouldn't be closer as any gene families, but we also did a pseudogene screening in the two genomes, and we found again way more in the genome of the symbiont compared to the genome of the free living species. Now, this is just to summarize potential drivers of the genome divergence in Symbiodiniaceae and in Symbiodinium, and also in their diversification. First, the extensive genetic diversity that we have in the family was attributed to sexual reproduction. However, this is just limited to homologous regions with high sequence similarity. Another explanation was were ancient events, for example, sea level changes, availability of hosts that would cause changes in the effective population sizes, and this has an impact on how genomes evolve over time. But here we're also proposing that symbiosis might be a big player in the genome evolution of this family and also in their diversification, because gene duplication contributes to changes in gene families, as we saw. Structural rearrangements cause loss of conserved synteny between the genomes, and the activity of mobile element further disrupts that synteny. They also impact gene structure and accelerate mutation rate. However, we then generated more genomes from the same genus in Symbiodiniaceae from genus Symbiodinium, and this is we also find them to be very different. Despite their lifestyle, whether they were symbiotic or free living. And something also very interesting that we found is shown in this tree. So this is a neighbor joining tree constructed based on distances estimated with shared and unique k-mers across the genomes, between the genomes. And we can see here that these two species that are actually free living species are the most divergent species in the genus, and they're also very divergent between themselves, which means there are also probably other mechanisms or forces driving genome divergence in the genus and probably in the family. And with that, I finish. I thank you for your attention, and I also thank all my team back in Australia, and also my colleague here at USF, John. And happy to take any questions.
Great. Questions? Yes, I will unmute you. Or actually, you can unmute yourself, please. Mawash?
Yeah. Okay. Thanks, Raul. That was a really interesting talk. Maybe you already mentioned this, but I'm just wondering, how closely related were these two species that you sequenced, or like how long ago did they diverge?
I didn't mention it, and I actually I don't show it. They're not too diverged. So I think they diverged about 5 million years ago. Yeah. So Symbiodinium is indeed the most basal lineage within Symbiodiniaceae. And we expect to see a lot of divergence within this genome. However, we didn't expect to see that much divergence, right? Yeah, but they still are classified within the same genus. And the more, if we compare the genomes from different genera, we see even more differences. So this still makes sense. Yeah.
Sorry, can I ask one more question? So these the different lifestyles, the free living, etc. Are they monophyletic or do they switch lifestyles in the
That's another issue. So in the genus that I was analyzing, as I mentioned again, is the most ancestral, and there is a monophyletic clade that clusters a lot of the free living species. However, in more inner nodes in the phylogeny of Symbiodiniaceae of the family, there are other taxa that are also free living. Actually, some genera are completely free living, exclusively free living, and some other symbiotic taxa have some taxa that are symbiotic. So this has happened like multiple times. Well, we don't really know what happened if it's that the free living lifestyle evolved multiple times in the phylogeny or the symbiotic lifestyle, right? That's something that we're interested in figuring out, but it's very difficult because the genomes are very, very different. Yeah.
Thank you.
No problem.
Claudio.
Yes. Hi, Raul. One question. I want to know if you look in detail at the contributions of different families of transposons, more specifically LTR retrotransposons, more specifically Gypsy versus Copia.
Yes, actually, most of those LTRs that you saw in that pic that I showed, that's mostly LTR Copia, and there is evidence in the literature from other groups that have shown that these have a play in adaptive responses like thermal stress. Yeah, in the family, not in the same species, but in the family.
So again, they were mostly Copia in both, right?
Yes. Yeah.
Okay. Thank you.
Yeah, no problem.
Maybe I'll ask one quick question.
Sure.
Can you? Maybe this is to speculate, but what would it look like if one of these symbionts, obligate symbionts, reverted to a purely free living lifestyle? What would be the first few effects that you'd expect to see in the genome?
I think I would see a lot of reduction of repetitive elements, reduction of all these duplicated elements. I think that's one of the things I would see. Also, another thing that you expect to see is a wider gene repertoire, like a more diverse set of metabolic pathways to respond to external stimulus. In the intracellular environment, you have a lot of redundancy with the functions of the host. So that would be some of the things. But that might take a while, I guess, for it to go back.
Yeah, because I was thinking, we seem to know a lot about the speed at which these things happen as you become a symbiont, but it's harder to study the speed at which they might undo themselves in the rare case that you actually manage to revert to being free living.
Yes, and there is a point in the transition to intracellularity where if you're too specialized to be a symbiont or a parasite, it's very hard to go back, right?
Yeah, of course. Yes.
So,
yeah, I don't think we're going to see many of those aphid symbionts return to being free living.
Exactly, yes.
Well, we're just about on time. We have one more question. Alistair. There. Oh, you, Alistair, you're not listening. You're not supposed to touch that. There we go. Alistair?
Can you hear me?
Yes.
We can, yes.
Yeah, sorry about that. I was just wondering, with the symbiotic Symbiodinium, do we know how extensive their external life cycle is? Because they have an external capability, so they're not sort of as obligately intracellular as some of the parasites that some of us used to thinking about.
Exactly. So that's something very difficult to assess, and that's something that we need to do to understand more how symbiosis can impact genome evolution of Symbiodiniaceae, of course. This specific type of parasite actually is not intracellular, but it lives in the interstitial space of the clams, mostly of the mollusks. But it's still kind of it's been shown that it's very associated with this organism. So it still kind of works as a facultative symbiont. So it's still kind of constricted. So what happens with intracellularity when with symbiosis and parasitism is that restriction that keeps them there, and the incapability to recombine sexually with other conspecifics, and that's what causes the changes in the evolution. And definitely having a clear picture of how much time they spend inside and outside of the host, and how viable are the life stages outside of the host, if they really reproduce sexually, would have an impact on how their genome change. Yeah. That's a good question. Yeah.
All right, thank you. So now we're going to have a coffee break for about 10 minutes. And this is going to be done by splitting everyone into 25 randomly assigned rooms. So there'll be 9 or 10 people in your room. You got instructions for how to accept this invitation, because it's an invitation. If you don't accept it, you'll stay here. But I'd like to encourage everyone to try it out. Maybe there'll be an old friend of yours in the room. Maybe you'll meet new people. We don't really know. But we're going to try it out and see how it goes. So I'm creating the breakout rooms now. And like I said, I'd encourage everyone to turn on your camera at that point and your microphone and just chat with whoever it is that you're linked with.